professor in the Department of Entomology and Forestry in the Natural Resources and the co-director of the Hardwood Tree Improvement and Regeneration Center at Purdue University. Matt is broadly interested in the chemical-mediated host colonization and mating behaviors of wood-boring beetles. The long-term goal of his research program is to develop effective pest management tactics targeting the chemically mediated mating system of beetles. This information will be used in establishing effective management strategies and programs, such by optimizing survey strategies, developing arboricultural techniques to bolster resistance, and improving techniques for detecting invasive species to improve the health, quality, and productivity of North American hardwood forests. Today, Dr. Ginzel will provide a general overview of the identification, impact, and management strategies for thousand cankers disease in the southeastern United States. And with that, I'm going to turn it over to Dr. Ginzel as I pass the baton here, which is always a bit clunky on WebEx. Okay. All right. Dr. Ginzel, you should be able to share. Looks great. Looks great. And we just needed to go full screen, and a reminder, you're still on mute. Okay, we can hear you.
Okay. All right, how's that?
That's great. That sounds great. Thank you so much.
You can see my first slide.
Yes, your first slide
Yes,
looks
great.
full screen and good to go. And if you could just scooch up to the microphone a little bit, you're just a little bit faint, but other than that, you look great.
Okay, how about now? Is that better?
Yeah, that's better. Thank you so much.
You bet, no problem. Thank you so much, Molly, for the invitation. And it's my pleasure today to talk a bit about some of the ongoing work that my lab's been doing with collaborators over about the last 10 years in thousand cankers disease. So today I'm going to sort of lay out a bit about the disease and what we've learned from our work here within the native range of black walnut, and end by talking about some management considerations for the disease. So TCD is a disease complex. So it's caused by a fungal pathogen called Geosmithia morbida, and it's vectored by the walnut twig beetle, Pityopteris junglandus. And when those two, when the beetle attacks a susceptible host, like black walnut, you see symptoms of the disease. So as the beetle bores into the host, it inoculates that tree with the fungus, and it causes a canker or a wound under the bark. And so those wounds grow to about the size of a nickel. And so with a lot of pest pressure, those wounds can coalesce and stop the flow of nutrients through the phloem, and the tree will succumb to the disease. Early symptoms of the disease are flagging of the leaves and yellowing, and then ultimately it can cause branch dieback and death. So it's named thousand cankers disease because in trees that are affected, oftentimes they find thousands of cankers. It's probably more aptly named tens of thousands of cankers disease. So this is really a disease that's death by 1,000 knife pricks, or tens of thousands of knife pricks. Tens of thousands of individual wounds caused by the beetle vectoring the pathogen into the host. So if you think about the disease triangle, where you have a pathogen, in this case, Geosmithia morbida, the success of that, the things that influence the progression of the disease are the survival of that pathogen, its dispersal, its host location, and the virulence. In this case, it's our understanding that Geosmithia morbida is a rather weak pathogen. And then on the host side, you have host susceptibility or resistance, and also suitability of this host. It seems as if most juglans are somewhat susceptible to thousand cankers disease, but Juglans nigra or black walnut being most susceptible. And then in the disease triangle, we have the pathogen and the host, and then the environment. So things like climate, wood moisture content, and biotic interactions of the fungi, other fungi and also with natural enemies, some fungi that are antagonists of Geosmithia morbida. But really, this disease doesn't fit neatly into the triangle. It's really sort of a quadrangle, because the survival of the pathogen is particularly dependent on a competent vector. And so a vector is a specific organism that transmits a pathogen. And in this case, it's walnut twig beetle. So we think about how we manage this disease. It probably will be most efficacious, best to manage the vector. If you manage the vector, you manage the spread of the pathogen. So this thousand cankers disease originated in the Southwest, and it spread throughout the West. And early on, in about 2008 or 9, we had a meeting here within the central hardwood region to talk about the threat of thousand cankers disease. And we were concerned about what would happen if thousand cankers disease was established within the East. Black walnut's one of the most valuable hardwood species that we have. And we noticed there was a lot of evidence and reports of black walnuts that were failing in the West. And so it did make it into the native range of black walnut. It was first found in 2010 in Tennessee, and then later in Ohio and Pennsylvania. And so that's about the time that my lab got interested. We focused on forest health issues. And so this was a big concern to us within the native range and to growers here in Indiana. And so we started establishing a trap tree study, where this was augmenting visual surveys that were done for symptomatic trees within the state. And also we girdled trees, and those we girdled them early in the spring. Then later in the fall, we felled those trees, cut them into bolts, reared the beetles out of them, and then looked for the presence of the pathogen on those beetles that we reared. Thankfully, we found no walnut twig beetles in Indiana. However, we did find some cocinine weevils that were recovered from 12 sites in Indiana. It's called Stenomymus pallidus. And we found Geosmithia morbida was detected in 3 individuals from 2 trees in an area in sort of southern Indiana called Yellowwood State Forest. And it was in a kind of abandoned walnut plantation, about a 30-year-old plantation. So that got us thinking, if this TCD model is complete, are there other potential vectors? If we go back to the disease triangle idea, are there other potential vectors in the disease? If so, it may be even much harder to manage. Also want to point out that we only recovered about 430 Stenomymus pallidus from walnuts that we had girdled at those 12 sites. So we girdled non-symptomatic black walnut. So that's got us asking questions about what beetles other than walnut twig beetle are capable of transmitting the pathogen. Does geographic location alter our current understanding of TCD? What influence does that environment, if you think back to the disease trial, play on the etiology or the progression of the disease? And do other pathogenic fungi also play a role? And so today I'm going to present some of the work that we did to answer a few of these questions. So there was an outbreak, as I mentioned, in Ohio. This is in Butler County near Cincinnati, Ohio. And this is a gentleman's backyard. Yeah, it's kind of a grove of walnuts and ash trees. And so we looked at these symptomatic trees, and this is work that I did in collaboration with Jenny Chuswick, who's a forest pathologist at the Northern Research Station in St. Paul. And so it's a nice project because it shows how these 2 disciplines of forest pathology and forest entomology can work together to answer important questions related to forest health. So we took from the symptomatic tree 2 30-centimeter segments from each of 4 branches. And then we took 8 30-centimeter sections from the main stem. And half of those sections, they were peeled, and we looked for the presence of cankers, and the other half we reared insects from, and then assayed those for the presence of Geosmithia morbida. So the samples were placed in these rearing buckets, and insects were identified and stored, and we identified all bark beetles and ambrosia beetles and weevils that emerged from these. So we recovered Geosmithia morbida from a variety of insects, primarily some ambrosia beetles, Xylosandus crassiesculus. 17 specimens were positive for Geosmithia morbida, Xylobrinus saxonii, and then also from some weevils. Again, Stenomymus pallidus was positive, 13 specimens of that, and Himatian errans. And so this was the first, this was the first evidence of Geosmithia morbida on any ambrosia beetles that are associated with TCD. And ambrosia beetles are a concern because they can attack weakened trees. I should point out that of the Stenomymus pallidus that we reared from our earlier study, there was only, I think I mentioned 400 and some. So if you think about that as it relates to forest health, it's probably not that big of a concern. If you think of, if each of those 430 beetles emerged from a TCD symptomatic tree and went to a new tree, they would really transmit a really small amount of inoculum. It would result in about 430 little cankers underneath the bark of a tree. Not enough to kill the tree, certainly. And so this shows some map and survey areas and locations of traps in 2015 to 17. And we did some detection surveys. States with reported trap surveys are highlighted in red: Minnesota, Illinois, Indiana, and North Carolina. States that were positive for TCD, Ohio, as you can see here in blue. And we used Lindgren funnels, trap trees, also in where you see the squares, we put down trap trees. And you can see in this red spot here is where there was a TCD location. And so all of the solid shapes show you that there's positive detection for Geosmithia morbida. So Geosmithia morbida was found all over Illinois and places in Indiana, and also in Minneapolis, and also a few areas in North Carolina. And this shows you some of the beetles. This is the insects for which we were able to detect Geosmithia morbida using a DNA probe or amplifying DNA, fungal DNA. And again, we add to the list. There's more ambrosia beetles, even some bark beetles, weevils, and powderpuss beetles. We even found Geosmithia morbida on some longhorn beetles. So it appears that Geosmithia morbida is more widely distributed than once we once thought. And so we found GM on the bodies of a diverse group of bark and ambrosia beetles. Although when we assayed for those, they weren't live propagules. We just found the presence of the DNA. And so they weren't in a stage that would allow for them to be alive and to propagate. And we found them in detection areas where there were no TCD symptomatic trees, which is alarming, but also in some ways it allowed us to posit that GM is probably more widely distributed than we thought. It might be just about everywhere. And so it really hammers home this idea that it takes a vector, a competent vector, and in the case of walnut twig beetle, it's an incredibly aggressive vector to cause the disease. So are these really vectors of TCD? Likely not. If you think about the disease progression, they certainly aren't affecting forest health. We don't find symptomatic trees. Even in Indiana, when we went back and we found that spot, yellowwood, we went back and surveyed for years looking for symptomatic trees and found none. There are no standing trees in Indiana that are symptomatic for TCD, although the pathogen is here. So I want to talk just a little bit about the difference between bark and ambrosia beetles. Geosmithia, or the walnut twig beetle, is a bark beetle, and they're often have a single host or a few hosts. They colonize weakened but living hosts. They feed on the phloem and cambium beneath the bark, whereas ambrosia beetles bore into the sapwood. And they're really fungus feeders. They're farmers of fungus. They carry fungus in these specialized pits in their bodies called mycotangia. And they can, because of that, they can attack a wide range of hosts. They really just need a place in which to grow their fungi. And they have their associations with fungi are a bit different. Bark beetles can be associated with a suite of fungal species, while ambrosia beetles are usually only associated with a few. And then the fungi assist in concentrating the macronutrients within the trees, whereas with ambrosia beetles, they're actually feeding on the fungus. Their immature stage within the tree, you can see here's the beetles, and this gallery is covered in fungus. They essentially cover the walls of the gallery with a combination of fungi and their own sort of spit, and they grow fungus. And so these little larvae here, white grubs, are feeding on that fungus. So we were getting reports of trees that were dying in Colorado, like this is a tree in Wheat Ridge, Colorado in 2010. And it was a rather sick tree, and it died. Whereas we were getting reports within the East that trees were able to shake the infection. This tree looks like it's symptomatic for TCD. You can see a lot of thinning of the crown. But yet some of those trees were surviving. They were coming back from the disease. And so we started a program in Ohio where we monitored the health of TCD symptomatic trees and surrounding trees, essentially determine the frequency of that decline. So is it spreading from a local epicenter, or is it that it stays rather concentrated? We did this in Butler County, Ohio. We went back to that place, the epicenter there, a gentleman's backyard, and also we went to our that plantation in Indiana. So the rationale for this is that monitoring these TCD symptomatic trees will provide a better understanding of factors that may affect the progression of the disease. So here are our field sites. As you can see, it's kind of a suburban area in Ohio. And the two areas are where there was the epicenters are right in here, here with the star, Avalon Station and Ashwood Knolls, which are two neighborhoods that are right adjacent to each other. And then we also set up transects in areas, in surrounding areas that had no TCD. And this is an aerial view of that plantation at Yellowwood State Forest, Brown County. So at each of these sites, we selected 40 walnut trees. We collected data on its history and stand and site conditions, tree size, precipitation, and the other species present. And then we went back and looked at the live crown on a monthly basis from 2015 throughout the growing season in 2015 and 2017. So we're essentially estimating live crown to the nearest 10%. And then we compared the rate of decline between the TCD symptomatic trees within that epicenter and neighboring trees, so other trees in the epicenter. And then we did a county by tree where we looked at the TCD epicenter or symptomatic trees within the epicenter to other trees outside in Butler County. And then we looked at the epicenter, so all of those trees, symptomatic and asymptomatic alike, with other trees in Butler County. And then finally, we compared the rate of decline between the epicenter, Butler County, and Yellowwood. And so this shows in this figure, this is the percent live crown on the y-axis, within the x-axis is years, and we're plotting the percent live crown on the top of the non-symptomatic trees in the TCD epicenter. versus those symptomatic trees. And we found really no change. So the symptomatic trees declined a bit, but not in a significant way. Whereas the neighboring trees within that epicenter, and sometimes these trees were just, 10 yards away, they hung in there. The crown condition did not decline. In fact, we had a little bit of a boost in crown condition from 2015 to 16. Similarly, when we compared outside the TCD epicenter in Butler County, they kind of hung in. They got a little shabby in 2018, but not due to TCD. And within the epicenter in Butler County, they hung in fine, and also Yellowwood. So we didn't see large-scale decline of these trees. Those trees that were affected with TCD, and we confirmed that they were infected by TCD, didn't get a whole lot worse. And what's interesting is if you look at the walnut twig beetle capture rates from 2013 to 15, in 13, there were 5 positive traps, most of them within that TCD, all of them rather, within that TCD epicenter in Avalon Station and Ashwood Knolls. And thousands of walnut twig beetles were collected from one trap in 2013 in that gentleman's backyard, right where we went. But in the following years, despite hundreds of traps being set, there were no beetles found in Ohio. So it appeared as if the beetles went away. And in the absence of that competent vector, we saw no progression of the disease. So our work showed that the disease didn't spread from neighboring trees, and there was no new development of disease. And TCD symptomatic trees had a lower percent crown rating, higher rate of deterioration in crown condition compared to the neighboring trees, but they didn't die, right? So it suggests that the disease was localized to individual trees, and there were no new symptoms or no symptoms appeared on new trees throughout our time. And the rate of decline within that epicenter didn't differ from other study sites in Butler County. So whatever rate of decline we saw was just the result of likely stress and other issues, other abiotic and biotic issues, but not from TCD. So this is a far different scenario than what we saw in the West. So in the Southwest is the native range of Juglans major. And at some point, the walnut twig beetle made a jump from Juglans major here to Juglans nigra in the east. And there's severe decline in mortality of planted nigra throughout the western US. Whereas in the east, Geosmithia is not found sporulating as frequently in walnut twig beetle galleries. So it seems as if that environment, if you go back to the disease triangle, the environment is having a big part of this, as well as antagonistic fungi. So there's a fungus called trichoderma that's antagonistic. It outcompetes Geosmithia morbida, and that can be found within the galleries of walnut twig beetle. So the disease development appears to differ between the West and the native range of black walnut. And I want to show you some evidence of that. This is TCD symptom change or recovery that was observed from 2010 to 2013 in Knoxville. So this tree in 2010 had about a 60% live crown rating, had moderate dieback branches. Whereas 3 years later, that tree has recovered. So there's new stem growth, an abundance of healthy foliage on this tree, until it reached about a 90% live crown. So this tree was able to shake the infection. And one reason that might be is some work by Griffin from Virginia Tech, and he did sort of a windshield survey and looked at the occurrence of the disease in Virginia and Tennessee, and found that when the precipitation was in Richmond, when the precipitation was low, there was new TCD progression. But when the precipitation was higher, it went into a kind of a quiescent state. The tree was able to rebound from that infection. And we look at in Knoxville from 2011 and 13, that time frame from the photo above, we see that there is no new symptoms of TCD observed, and it was associated with the growth and recovery of Niagara. So it suggests that the environment has a big part to play. And so ecological factors like the environment, and so there's these climatic differences between the expanded range and the native range of eastern black walnut could affect the growth and dispersal of GM. And it could do that by modifying the suitability for infection and also canker development in the bark, and influencing its competitive interactions of the pathogen with other fungi. So I wanted to spend a little bit of time talking about some work from one of my graduate students, Jeff Williams, who you can see here in the upper right. He just graduated recently. He's doing some postdoctoral work with the Forest Service in Eugene, Oregon now. And so he looked at fungal competition and moisture content in wood. And so moisture content is a function of temperature and humidity. And so we collected naturally infested material from a site in Walla Walla, Washington, and cut those into discs. And then placed those discs in environmental chambers where we could alter the humidity of them using saturated salt solutions. And so then from that, he calculated the final moisture content and then observed the growth of the fungi on those wood discs after 100 days. So he brought them all down to varying levels of moisture and then allowed them to grow, the fungi to grow, and looked at that growth and the projected survival of GM based on those data. He then modeled the survival of GM using models of moisture content and also of climate. And here are some of his data where this is the survival rate on the y-axis, the moisture content along the x. And these are fungi that were also present with GM. And what you find is that at higher moisture content, Geosmithia morbida gets outcompeted. Its sporulation depends a lot on moisture content. Sorry, this is just moisture content. That's just competition. This is just survival rate. So its sporulation depends on moisture content, and it competes and sporulates at low moisture content, less than 10%. But when GM is out, when moisture content rises above 20%, GM is outcompeted by other antagonistic fungi, like trichoderma, this one here, and other aspergillus fungi and clonostasis fungi. It's also interesting is in the sterilized wood, where there were no other fungi present, GM did just fine at all moisture contents. So it's really the moisture content that's dictating this competitive interaction with other fungi. So from that, we modeled this using GM survival and TCD risk projected with moisture content. And so in the Western US, survival is expected to be high. You can see the green areas are higher rates of survival. And as we get into these cooler colors, it's lower rates. And then we modeled it, and in the East, it's expected to be quite low. Although I don't know if you can see, there are some areas like in Tennessee where there was TCD, where it's expected to be a little bit higher. So this model did a nice job of reflecting where TCD has been an issue. And then if you look into the Southeast here, in your area, it looks like it's historically rather low. And in climate scenarios with low emission in the next 10 years and also 50 years, it also appears to be somewhat lower. But with higher emission rates of CO2, TCD may expand into the eastern US. You can see here under high emission CO2 in 50-year time frame. So in summary, there are many beetles and weevil species that are found with Geosmithia morbida DNA on their bodies. Though few had live propagules, and some detections were in states with no known TCD. TCD symptoms occur during times of below average precipitation, and then trees are able to recover with more rainfall. TCD doesn't progress in the absence of the primary vector. We think that these connections with ambrosia beetles, other bark beetles beyond walnut twig beetle are just casual in their relationship. They pick them up and they move them, perhaps it might cause a canker, but it's certainly not, it doesn't transfer enough of Geosmithia morbida to affect the trees. It appears that if mass attack isn't followed by subsequent attacks in following years, that the host response may indeed prevent the further development of cankers in the absence of walnut twig beetle, like we saw from our work from Ohio and Indiana. So our findings support the hypothesis that these other beetles have little impact on disease severity and spread, and that Geosmithia morbida appears to be adapted to dry climates, low substrate moisture, and survival is expected to be rather low in the east, which is encouraging for those of us who are interested in the sustainability of walnut on our landscape. So I wanted to end with a bit of a talk about some integrated pest management work for TCD, and then allow some time for questions. I don't know, Molly, if there's questions now, if you'd like me to stop or just keep going.
We actually haven't gotten too many in yet, so I encourage folks, please don't hesitate to write in. And yeah, Dr. Ginzel, this is going great. Please just go ahead and proceed if you don't mind.
We're kind of switching gears a little bit, so I thought if there were questions up now, I could answer them. That's no problem. So I wanted to switch gears, and we went to an area in the West. This is in Walla Walla, Washington, where there are plantations of black walnut. So the Hardwood Tree Improvement Regeneration Center at Purdue is a collaborative venture between the U.S. Forest Service, Northern Research Station, and Purdue University. Its mission is to advance the science of tree improvement, management, and protection, with special emphasis in the central hardwood region. And so we've been breeding black walnut at Purdue for growth and form for over the last 50 years. So we have some improved walnut, and we've planted these in out in Walla Walla with the cooperation of some of our stakeholders there. And so this is part of that property. This is Bart Nelson's property, and I'm going to show you some photos of his plantations. And so what we want to do is compare the response of walnut twig beetle populations at 3 walnut plantations using, essentially we took an IPM program and split it into constituent parts, and then evaluate each of those together. So sort of 3 different approaches, and compare the colony response to different chemical treatments over time to manage walnut twig beetle. So there are 3 plantations here, and so these are part of, as I mentioned, our breeding program. And so the first is Cottonwood Creek, and 1 plantation went in 2001, and 1 went in 2009. So that 20-year plantation is pretty large at this point. This shows you the 09 plantation, so that's what you get from 12 years of growth. In Washington, the trees do really well in that climate. And the other had a similar sort of setup with a 2001 planting and 2009 planting at Yellowhawk Creek. And the other is at Russell Creek in 2009. All of this work is predicated on our ability to monitor walnut twig beetle populations using pheromone-baited funnel traps. So this is a Lindgren multi-funnel trap. There's a commercially available pheromone lure here. It's can be purchased for land managers and others. And so when we first learned about this disease was 2016, we learned from the landowner that walnuts were looking kind of bad. We knew walnut twig beetle and thousand cankers disease was in the area. So we went out to assess the problem. And so the first thing I did was to put up some of these multi-funnel traps to see if I could detect walnut twig beetle. And I certainly did. The way I had full-blown thousand cankers disease within this plantation and the other 3. So monitoring occurred for 12 to 16 weeks during the late fall, late summer, early fall. So Walnut twig beetle has sort of two flight periods, has an early spring flight period and then a late fall flight period. And in most places, that late fall flight period is stronger. There's more pressure in the late fall than in the early spring. So this gives you a little bit of history of that Cottonwood Creek. This is the 2009 planting. It's called the Mickler clone test. So this is grafted clones versus select seedlings from our breeding program. About 420 trees went in. And then we have a provenance test where we have seedlings from Indiana, Tennessee, and Oregon that was put in 2001. And so in 2008 is when TCD was confirmed in Washington State. We did all of the grafting for the Mickler plot there in a greenhouse on site that year. And 2009, it was confirmed in Walla Walla. Larry Malott at Whitman College was first observed those symptoms. And then the Mickler plot went in. We weren't really that aware of the problem in 2009. There was some thinning that happened in 2012, and it was in 2013 that the first symptoms, now that in retrospect we find out, appeared in these really large, there's 25 seedlings that went in 1980. So went in these, what we call the old trees in this southeastern corner. So it's 2016 is where we confirmed TCD in this area, and also in the provenance test and a few, some beetles in 2009 planting in that Mickler plot. So we collected an average of about 111 walnut twig beetle per trap per week throughout that late season in 2016. So we thought, yeah, it's a bit of a moderate infestation. And you can see some of the symptoms here. Some of the leaves are starting to see some branch dieback and yellowing and leaves in the upper canopy. The other spot is called Yellow Hawk Creek. And so this planting also has a provenance test that went in 2001. And there's about 380 walnuts from our breeding program. A lot of them here, which is a bit of a mixed planting as well. It's not all walnut. And so in 2001, there was, in this 2001 planting area, we collected almost 1,600 walnut twig beetles a week. And in 2009 planting, around in the 30s. So it's far more advanced in this area. And you're wondering why that might be. And I want to show you some of the pictures here. This is Jim McKenna, who's now since retired operational tree breeder with the Forest Service. And you can see these trees are in advanced stages of TCD. And also, you can tell by looking at the picture that the plot map looks a lot different than what it did when we had this aerial photo taken. There are trees missing. And so we met with the landowner and said, you know, you have a serious problem here. And you can see here is some, these trees, he's been cutting some trees down. So we met with him to talk about what options we could do to manage this disease. And we also found in the corner that he'd been piling all of those dead trees up in the corner of his property. And so essentially he was, had a walnut twig beetle rearing program. And so, you know, as a cultural control method, our first recommendation was to get rid of this pile of wood, like burn it, get it out of here, you know. But a lot of these trees, they ended up dying rather quickly from the disease. And then the third site is called Russell Creek. And so TCD symptoms were most advanced in some of these older trees. Like there was a big walnut here, and there's a creek that runs through here, and there's some, you know, 100-year-old walnuts along that creek. So our thinking was they were coming from there. And so we also encouraged him to take this tree. You can see the crown of this tree is already somewhat gone in 2016. And there are more symptomatic trees here than any other location. We call this a high infestation rate. Average about 1,300 walnut twig beetles per week in that area. Get more shots of some of those symptomatic trees. And so this is We peeled the bark of some of those trees at Russell Creek, and you can see the cankers here, these little black cankers that are caused by the fungus. And so we flagged this tree as a tree that had was confirmed for TCD. We took some samples for assaying for the pathogen, but there's really not much hope for this tree at this point. So you can kind of see sort of the density, the competency of this vector of the walnut twig beetle in areas where there's a lot of disease pressure. So, for example, this is one night's capture in one area, and this is 1 week's capture. And so we started first counting these, and these are really tiny beetles. You can see how small they are relative to, they're about the size of a printed period on a page. And so then we decided, we better start developing a scheme to subsample these, because no one is going to count this many beetles over the course of a week. So what we proposed was a site-specific IPM approach. So at Cottonwood Creek, where there was a moderate initial infestation, we were going to use girdle trap trees. We knew from our other work that beetles are attracted, ambrosia beetles and bark beetles are attracted to girdle trees. So our hope was that we would take girdle trees and apply a stem girdle that we then treated with glyphosate to the tree in late summer before that fall flight period. Spray the trunk of that tree with bifenthrin, so we'd get a little bit of contact insecticide. And then trees that we called crop trees, which were those of the best form, we wanted to see if we could protect those with trunk injected insecticide, emamectin benzoate, which is effective, really effective against emerald ash borer and also other bark beetles. And then, so that was one treatment, emamectin benzoate alone. We also did emamectin benzoate and propiconazole together. So propiconazole is a fungicide, so our thought was, okay, perhaps a fungicide would help in killing the pathogen, and we compared the crown condition of those treated trees to untreated controls. At Yellowhawk Creek, where the high initial infestation in the older trees and lower in the 2009 planting, we just chemically treated crop trees only, so we didn't use the girdle trap tree approach there. Whereas in Russell Creek, we had the girdle trap trees only, so we could compare each of these components. So the trap trees were girdled in the fall before the flight period in 2017 through 2019, and they were removed during the dormant season, February or so, from Cottonwood Creek and also Russell Creek. And then we applied a single rate chemical treatment in 2017 to crop trees from Cottonwood and Yellowhawk. And we used ArborJet. Material was donated from ArborJet and from Rainbow Tree. We used ArborJet system. And so this is a tree IV system. As you can see, what happens is you drill a hole into the base of the tree and inject a port, plastic port. That active material then goes into this canister. It's pressurized, open the valves, and it feeds into the tree and gets taken up into the canopy. In 2018, we also tried a double rate. We tried a double rate of emamectin benzoate and a double rate of propiconazole. And then we did crown ratings, canopy ratings to the nearest tent each summer, 2017 to 2020, to look at to determine the frequency of crown decline within those treated trees relative to the controls. So I just want to show you some data around beetle capture. This is each of the sites. Cottonwood, we essentially combine those two, whereas in Yellowhawk, we kept those separate. And you can see there are a lot of beetles at Russell Creek. That's the spot where we use the trap trees alone, and in Yellowhawk initially. But there was a big decline at all sites after 2016. At Yellowhawk, where there were no trap trees, it exceeded other sites in beetle capture in 2018 and 19 through 2020. So what it shows is that the girdle trap trees worked. They worked to concentrate beetles to these girdle trees They would colonize the tree and then be removed during the dormant seasons. Girdle trap trees seems to work rather well. And we also found that in the emamectin benzoate, yellowhawk, in those areas, that after 2 years after injection, you start to lose control, which isn't surprising. Those trees put on new phloem, and so they're no longer, the material is no longer accessible to the beetles. So this shows you some of those canopy ratings at Cottonwood Creek and Yellowhawk relative to the untreated controls. They kind of hung in. We saw an initial boost from emamectin benzoate here, and propiconazole didn't seem to help a whole lot, and they kind of hung in throughout the time frame. So all comparisons, emamectin benzoate alone exceeded or didn't do any worse than all other treatments. So it provided some protection against canopy decline. But the double rate was really bad. In fact, there were some trees that the landowner was concerned about the following year after, in 2019, after that application. And he said, I think these trees have TCD. And I tell him, no, I think it was me. You had MDG instead of TCD because of, I think it's just, we saw a lot of toxicity from the propiconazole. So you can see wherever we have the double rate of propiconazole, tree decline is a lot. Double rate of emamectin benzoate kind of hung in relative. So what we see in the crown condition for untreated control trees compared to all sites didn't differ in the 2019 planting. The crown condition of the untreated control trees in Cottonwood, those where we use trap trees, exceeded those of Yellowhawk Creek, where there were no trap trees. So it looks again like trap trees are a benefit. So I wanted to end by talking a little bit about some management considerations with this work. And one of them is stand monitoring is critical to success. We have a tool available for us to do detection and monitoring through these pheromone-baited multifunnel traps. And the removal and destruction of symptomatic trees can help. So those are trees that we, if they were symptomatic, we chose those as the girdle trap trees. And it helped to minimize the spread of TCD to other trees. And also, girdle trees can be a really useful way to maintain and reduce walnut twig beetle populations. Essentially act as an attractant, you get them out of there in the winter. It appears that emamectin benzoate can be effective in protecting walnut from TCD. The benefit wears off after a few years, which isn't surprising. So in some might suggest reapply. And combining trap trees and the crop tree trap tree methods, trap tree and crop trees in an integrated approach, that's chemically treated crop trees, offer potential to reduce walnut twig beetle populations and protect these high value plantations from TCD. So with that, I have a lot of folks to thank. Jenny Jeswick in the U.S. Forest Service, Fred Miller, who's at the Morton Arboretum, folks that are associated with Jenny or Paul Castillo and Melanie Moore, and then folks at the IDNR, Phil Marshall and Vince Burkle, Bart Nelson for his access to his sites there in Walla Walla, Don Grossman, Rainbow Metro Parks, Butler County, who helped us with our work there, and also OSU's Extension Office, and then a lot of students, technicians, instrumental in this work. And funding came from the Forest Service through a Forest Health Evaluation Monitoring Grant, also through FHP. I'd like to thank Purdue and Indiana DNR also for funding. So with that, I have some time for questions. I'd love to take some. Thank you.
All right. Thank you so much, Dr. Ginzel. That was such an excellent presentation. Lots of new, great information that I certainly wasn't aware of. So yes, we're now going to open the presentation for questions from the audience. If you have yet to type your questions into the chat window, please do so at this time, and I'll do my best to get to them. Although we did get quite a list stacked up. So I guess we'll just dive in. Thank you, Dr. Ginzel. Is Geosmithia morbidea native to the East Coast, or did it come with the beetles? Does that make sense?
Oh, I'm muted. Sorry.
Oh, okay. Sorry.
I didn't have my video on that time during that time. I'm sorry about
Oh, yeah, neither did I.
that. Okay. I was like, I didn't see what's. Could you repeat the question for me? I was playing around.
Yeah, someone was just wondering if Geosmithia morbida is native to the East Coast.
No, so it's native to the southeast or southwest. I always get those two mixed up. It's native to like New Mexico, Arizona, that kind of area. So at some point, and so is the walnut twig beetle. And so it seemed like at some point these two made an alliance and were able to hop onto black walnut, and then it spread from there.
Okay, that already tackles another question that we had. The geographic origin of both walnut twig beetle and Geosmithia morbida are the southwestern U.S.
Yeah.
Yeah. Okay. In your opinion, what makes walnut twig borer such an ideal vector?
Yeah, and it's questions that we've been addressing currently are. So it seems like there's just not much resistance. So when you look at little walnut, there seems to be some resistance to it. So there's some host plant resistance, anti-cyanosis, antibiosis sort of things that we're trying to uncover now as to why are these trees so susceptible to attack by walnut twig beetle.
Fair enough.
And it could be that Geosmithia morbida is helping in that regard. I mean, we don't know. We don't know if they're what sort of relationship they have with that. Are they eating? Some suggest perhaps they're feeding on it. But we don't know.
Yeah. Can you offer advice for sampling trees for thousand cankers disease?
Yeah. So that's part of the problem, right? Because they look like they're just in decline. Like when Colorado, when there was first symptomatic trees, they thought, oh, maybe it's some kind of anthracnose or something. And so the best way to sample them is to look for those symptoms of flagging, yellowing, brown dieback, and then start to peel the bark a little bit, and you can see the symptoms. But where you have GM, you will have a lot of walnut twig beetles. So I think the best way to do it is to put up a pheromone bait trap and see if you start catching.
There you go. There you go. Okay. Yeah, we got a couple questions like that about how to sample quickly and easily in the field, and that sounds a good place to start, just so you can discern whether you have the beetle populations available. Okay. Someone said my grad research is looking at the economics of black walnut plantations in West Virginia.
Fantastic.
Would you discourage landowners from establishing a black walnut plantation in West Virginia due to thousand cankers disease, or just advise caution?
Yes. Yep.
Yes.
I'm all in on black walnut. That was part of our concern. So part of why we did this work, black walnut is huge in Indiana. And so there were folks who have there are a lot of black walnut plantations. People put in small plantations, they might have an acre here or so of black walnut. And in the fear of thousand cankers disease, timber buyers going around saying, hey, now's the time. You better sell now. You harvest now. And so a lot of that, my work was generated from that idea, is to say, no, don't. Put the chainsaws away. It appears like TCD is not as severe here within the native range of black walnut than it is in the West. So yeah,
oh, that's interesting.
Discourage growers from continuing to cultivate black walnut.
Yeah. She was wondering, what recommendations would you have? Would you just let them know to monitor, keep an eye out?
Yeah.
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 Okay.
Stand monitoring is critical. So there's a group here in Indiana. It's called Arbor America. It's a Spanish company, and they bought up about 640 acres of prime Indiana farmland, and they planted it. In fact, that picture that I showed you is their plantation. If I can show you, screen sharing it. But they've planted a lot of HTRC black walnut, and this is a multinational company with lots of investors, and so they're of course really concerned about black walnut, and it's highly cultivated, fertigate, all these sorts of things. Do a lot of pruning, and so we started in my lab. We do a little bit of a monitoring program each year, which has been really great. It's giving my students an opportunity to work with with landowners and get involved in some of this monitoring. And they were really concerned about it, but I think the best thing to do is just be vigilant,
Yeah.
do the monitoring, and look for signs and symptoms.
Okay. Okay. And install those pheromone traps. Couldn't hurt.
Yeah, that's what we've been doing. Yeah.
Okay.
We don't catch much,
That's good advice.
but yeah.
Gosh, we got a lot of them coming in. Do you still have a few minutes? Oh, we're closing in on the
I can
hour.
see them. I have time.
Are they coming through? Well, I got them all. They're just coming in and I haven't even finished this list yet. But I'll try and summarize the ones that we're getting doubles of.
Okay.
How do other species of juglans react?
Yeah, so we know that Regia doesn't do as poorly as Niagara does. We're doing some work now looking at some of the hybrids. So we have hybrids here of Antifolia, of Cinerea, butternut, little walnuts doesn't react as bad. It seems that Jugland Niagara wingnut shows some susceptibility. So there's some Pterocaria that shows some susceptibility, but it looks like black walnut is the most susceptible by far.
Okay. Someone was wondering if the chemical control strategies that you mentioned could be used on trees where nuts are collected for consumption.
Yeah, so we've been doing that work. Yeah. So
Nice.
what we did is we injected a bunch of trees. All right. Some of the trees here, we did some work here in Indiana. This is work with Phil Lewis at Otis.
I think you might have frozen, or at least maybe that's just on my end. Oh, looks like we might have lost him. Stay tuned.
Oh,
okay, you're back.
I'm back.
Just internet connection stuff.
Maybe I'll stop.
Yeah, you're back.
Yeah, maybe I'll stop my video. Would that help?
Yeah, it's worth a shot.
Okay. So yeah, we did this work where we injected trees and then looked at residues, both emamectin benzoate and imidacloprid in the leaves. Think about a walnut; it's like has a shell around it, right? So we looked at the shell, the husk, and in the nut. And what we found is we found residues in the leaves, not much in the hulls, nothing in the nut. It's on label for walnut, twig beetle, and walnut, but there is a concern because it's edible.
Okay. Sorry, you were a little bit in and out on that last one, but I think I gotcha. And we'll have in the recording, folks, if you want to tune back in, we'll have subtitles too, so that'll help. Let's just finish up with I think just one more. Well, we'll do one and one quick one. Are there any other beetles in the West that have the Geosmithia morbida fungus?
I haven't looked.
Sure.
I don't know of any reports of that, like the reports coming out of the East with the ambrosia beetles and things. I think it's probably because there was such a problem that, there's so much of the vector itself that they weren't concerned about alternate vectors. But so I don't know that part. I'll tell you that we collect very few beetles in Walla Walla beyond walnut twig beetle.
Okay. This is an interesting one. Okay. So someone said here in Southern California, we had quite a few black walnuts, Juglans californica die, but from the stumps, new sprouts emerged, and now these are multi-stem 4 to 5 inch DBH trees. How viable are these trees? Does GM persist for many years, especially considering that they are native and protected in many jurisdictions?
So how viable are the trees? In what way are they thinking there?
I guess they just mean maybe vulnerable.
Okay.
Do they currently contain the fungus?
So probably not. GM will survive really well at low moisture content. So it'll be there. It's in the area.
Right.
Now, the extent to which the beetles will attack those trees, unsure. But I would suspect that, if they were hit once, they might hit again.
Okay. Can you do one more? Do you mind? One more just came in. Okay. So in this effort to control walnut twig borer, is it because of the damage from walnut twig borer, or is it to control it as a vector for GM? I thought we just demonstrated that many beetles act as vectors and that this disease is largely site, soil, or climate influenced more so than by walnut twig borer. Thanks in advance.
Yeah, so those other insects probably aren't competent vectors, for one, right?
Okay.
It's not going to
Right.
tree health. Then the first part was, can you repeat that part for me? I'm sorry.
Oh yeah. It's basically, why are we targeting WTB? Is it because of the damage from, or is it primarily because it is the primary vector for
Yeah,
GM?
it's because it's the primary vector primarily. So, you know, as far as wood quality goes, these beetles are underneath the bark. They don't really affect wood quality per se, but until they end up killing the tree, and then it's, you know, future wood quality, you know, it's future growth. So yeah, we've been doing work. I did some work with Jenny Juswick and Ron Mack and some folks at Virginia Tech, Marshall White, where we developed a vacuum steam treatment for saw logs. So, you know, so for export. So this is vacuum driven steam, and we could kill the pathogen and the beetle with vacuum steam, so that, you know, that way we wouldn't have to do, you know, other kind of treatments. So that's kind of cool work. But yeah, primarily the idea is, you know, is this, I think the question is this a pathology problem or an entomology problem? And I think it's an entomology problem, because the pathology is such that the pathogen is terrible. It's rather weak on its own. You know, it's not enough to kill the tree. And it causes what we think is just an annual localized canker. This isn't a systemic kind of thing. You know, it causes one little wound, but if you have one little wound multiplied by tens of thousands of beetles, that's the problem. So I think the way to manage it is to manage the beetle.
Perfectly put. That's a great place to stop. Thank you so much, Dr. Ginzel.
Thanks so much.
That's great. That's great. We really appreciate you being here. Folks, I will stick around for about another 10 minutes or so. We're going to cut Dr. Ginzel loose. We've definitely I've taken up more than enough of your time. But I can't thank you enough.
Yeah, and if folks want to contact me, can you put my email address in?
Certainly. Yeah, appreciate that. I'll throw it in the chat box.
Sounds great. Thanks.
Okay. Thank you so much. You have a good rest of your day. We really appreciate you.
All right. Take care.
Bye-bye.